Welcome to this overview of transcriptomics and RNA sequencing. In this presentation, we'll explore how RNA-seq reveals gene expression patterns, the technologies behind it, key analysis steps, advanced applications, current challenges, and future directions. Think of this as a guided tour through the methods and insights that let researchers peek into how genes behave in cells and tissues. This slide outlines our roadmap. We'll begin with an introduction to transcriptomics, then cover RNA-seq principles, data analysis, and advanced applications. After that, we'll discuss practical challenges and emerging trends before finishing with a summary and outlook for research and clinical use. This structure will help you connect methods to biological questions and real-world opportunities. Now we begin the introduction to transcriptomics. I'll set the stage by explaining what transcriptomics studies, why it matters for understanding cellular function, and how RNA profiling gives us a dynamic view of gene activity across conditions and stages. This opening frames the rest of the talk so you can see how each method and analysis fits into the bigger biological picture. This overview summarizes what transcriptomics is and why it's useful. Transcriptomics examines the full set of RNA transcripts to reveal gene expression patterns, regulation, and functional changes across conditions. That insight drives discoveries in molecular biology, helps identify disease biomarkers, and supports drug target research, so it's central to both basic science and translational medicine. Before diving into RNA-seq, let's cover RNA basics. Cells contain many RNA types including mRNA, tRNA, rRNA, and regulatory non-coding RNAs, each with distinct roles. DNA is transcribed into RNA by polymerases, and RNA molecules undergo capping, splicing, and polyadenylation, which shape stability and translation, influencing the transcriptome we measure with RNA-seq. Next, we'll look at the core principles behind RNA-seq technology and how experimental design influences the data you get. This section explains the lab workflow, the sequencing platforms available, and the quality controls required to generate reliable transcriptome profiles, so you can appreciate both the opportunities and the practical constraints of the method. The RNA-seq workflow starts with careful sample preparation and RNA extraction to preserve integrity and reduce ribosomal contamination, followed by library construction where RNA is converted into a sequencing-ready format with adapters and amplification, and ends with sequencing on platforms chosen for read length and throughput depending on experimental goals. Generating sequencing data requires rigorous quality control at every step. Read quality assessment checks for base calling accuracy and biases, adapter trimming removes technical sequences and low-quality fragments, and handling technical variability through normalization and replicates is essential to minimize batch effects and ensure robust downstream conclusions. Now we move into data analysis and interpretation where raw sequencing reads become biological stories. This section covers how we align reads to references, assemble transcripts, quantify expression levels, and apply statistical frameworks to extract meaningful patterns, all while keeping an eye on artifacts and validation to ensure biological relevance of the results. Read alignment and quantification are the backbone of RNA-seq analysis. Reads are mapped to a reference genome or transcriptome to locate transcripts. Transcript assembly reconstructs isoforms and alternative splicing, and expression quantification counts reads per gene or transcript, providing the measurements required for downstream comparisons and functional interpretation. Differential gene expression analysis identifies genes that change between conditions and relies on robust statistical models. Methods like DE-seq2 or EDGER model count data, control for variability, and report significance, while visualization tools such as heat maps, volcano plots, and PCA help interpret patterns, cluster samples, and highlight biologically meaningful changes. In advanced applications, we use RNA-seq to probe complexity beyond bulk expression, exploring isoforms, cellular heterogeneity, and regulatory networks. This section highlights applications that push methodological boundaries and demonstrate how deeper transcriptome profiling can reveal new biology, from splicing dynamics to cell type-specific signatures and disease mechanisms. Alternative splicing analysis uncovers how a single gene can produce multiple transcript variants. By detecting exon-exon junctions and quantifying splice forms, we can link splicing patterns to proteomic diversity and disease processes. Specialized tools and pipelines make it possible to detect novel events and compare splicing across conditions, adding an important layer to transcriptomic interpretation. Single-cell RNA-seq provides high-resolution views of cellular heterogeneity but brings unique challenges like low RNA input, dropout events, and complex data processing. When done correctly, it reveals distinct cell types, states, and trajectories within tissues, and analysis strategies include normalization, clustering, dimensionality reduction, and trajectory inference to reconstruct dynamic biological processes. Next, we'll consider common challenges and practical considerations that can affect RNA-seq studies and their conclusions. Understanding these limitations helps design better experiments and interpret results cautiously, whether the issue is technical bias, insufficient depth, or biological confounders, and highlights why rigorous controls and replication are crucial. Technical limitations can skew RNA-seq results if not addressed. Sequencing bias and base calling errors may distort transcript representation, and uneven coverage or low sequencing depth can miss low-abundance transcripts or alternative splicing. Being aware of these constraints helps guide protocol choices and sequencing depth to match the biological questions being asked. Biological and experimental factors are equally important in shaping transcriptomic data. RNA integrity, sample handling, and storage affect results, while batch effects and inadequate replicates can introduce non-biological variability. Careful experimental design and awareness of interpretation caveats are essential to distinguish real biological signals from technical or sampling artifacts. Looking ahead, transcriptomics continues to evolve with new technologies and integrative approaches. This section previews emerging sequencing technologies and the move toward combining multi-omics data to gain richer, system-level insights. The future promises higher resolution, spatial context, and better integration with proteomics and epigenomics for deeper biological understanding. Among emerging technologies, long-read RNA sequencing and spatial transcriptomics are transforming how we study the transcriptome. Long reads capture full-length isoforms and complex splicing, while spatial methods map expression back into tissue architecture, enabling precise localization of transcripts and a more complete view of gene regulation in health and disease. Integrative multiomics combines RNA-seq with proteomics, epigenomics, and metabolomics to link transcript levels to protein abundance and regulatory modifications. This systems biology perspective helps unravel complex networks and mechanisms by integrating complementary datasets, offering a more holistic understanding of cellular function and supporting robust biomarker discovery. We end with a summary and conclusions to tie the main messages together and reflect on practical implications for research and clinical work. RNA-seq has revolutionized expression profiling, yet its full potential depends on rigorous experimental design, careful analysis, and thoughtful integration with other data types to translate findings into real-world impact. The key takeaways are that RNA-seq provides sensitive, quantitative insights into gene expression and regulation, but comes with challenges such as cost, data complexity, and sensitivity for low-abundance transcripts. Appreciating both the strengths and limitations enables researchers to design better studies and extract biologically meaningful conclusions from transcriptomic data. Finally, the outlook for RNA-seq in research and clinical settings is promising. Transcriptomics offers translational potential for biomarker discovery, personalized medicine, and improved diagnostics, while innovations like single-cell and spatial approaches will continue to enhance resolution and open new possibilities for precision medicine and biological discovery. Thank you for following this overview of transcriptomics and RNA-seq. I hope this presentation clarified key concepts, workflows, challenges, and future directions, and that it inspires you to think critically about applying these tools to your own research questions and clinical goals.